Welcome to the Breakthrough Broadcast, a podcast about innovative STEM solutions to real-world problems. From emerging technologies to practical research, Drew Reck explores how science, engineering, and bold thinking are shaping the future.
Hi, I'm Drew, and in this episode, we're exploring a technology that pushes the boundaries of what humans can see. Three-Electron microscopy. It's a method that reveals the inner architecture of biological tissue with a clarity that didn't exist until just a few years ago. Instead of relying on a single type of electron signal, or even two different types, this technique collects three distinct electron interactions from the same sample and merges them into one unified, highly detailed image. The result is a view of the microscopic world that is rich, layered, and scientifically transformative. Electron microscopy didn't start as a highly optimized scientific tool. Its earliest form appeared in the 1930s, when researchers were first realizing that visible light has a physical limit that prevented microscopes from seeing anything smaller than bacteria. Scientists needed a way to break past that limit, and electrons, with their increased capabilities for precision, became the solution. Early prototypes were bulky, unstable, and notoriously hard to operate, but they opened an entirely new window into the microscopic world. For decades, electron microscopes improved slowly. Engineers refined vacuum systems, invented stronger electromagnetic lenses, and developed better detectors. But even then, these machines could only create images from a single stream of electrons. That meant samples with uneven textures or mixed materials produced incomplete or distorted results. During the second half of the 19th century, researchers kept pushing for ways to get more information out of each sample, but the technology simply wasn't ready. What changed everything was the shift to digital imaging and advanced computation. Digital detectors allowed scientists to capture electron signals with extraordinary precision, and powerful processors made it possible to reconstruct complex structures in ways that were previously impossible. These advancements laid the foundation for multi-signal imaging, where different electron interactions were collected and combined. Three-electron microscopy grew out of that evolution. Instead of relying on one incomplete signal, it brought together multiple electron behaviors to produce an image that was richer, clearer, and far more accurate than anything before it. To understand why this matters, imagine examining a thin human slice of tissue under a traditional microscope. Even with careful preparation and high magnification, certain structures blur together. Layers overlap, tiny membranes hide behind denser material, and deeper features disappear entirely. Scientists have always been able to see parts of the picture, but those parts often lacked context. Three-electron microscopy was designed to solve this. By capturing three completely different electron signals, it reveals surface features, internal organization, and compositional differences simultaneously, turning what used to be fragments of information into a continuous, interpretable picture. Before diving into how the method works, it helps to understand the foundations of electron imaging. Electron microscopes function by directing a beam of electrons at a sample. When the electrons interact with the atoms in the tissue, they behave in several possible ways, depending on the energy of the beam, the structure of the material, and the arrangement of the electrons inside the atoms that are being hit. Some electrons are ejected from the surface of the tissue after being hit by the electron beam. Some reflect backward after colliding with the tissue, and others pass through the sample altogether. Each of these interactions carries different information about the material when the electrons eventually return to the microscope. Traditional systems usually relied on only one of these interactions, or, in more advanced setups, two. When we incorporate three, this allows the microscope to detect the surface, the density, and the internal layers of the sample, all in one go. The three signals involved are called secondary electrons, backscattered electrons, and transmitted electrons. Secondary electrons are those that originate from the surface. They're low-energy particles released when the incoming beam knocks outer electrons loose from atoms in the sample. Because they escape from only the top few nanometers of your sample, they highlight extremely fine details on the surface, like textures, ridges, folds, and the contours of membranes. Secondary electrons are the reason scanning electron microscopes, or SEMs, are known for their crisp, sculpted appearance. Backscattered electrons reflect something different. These are high-energy electrons from the original beam that bounce back after interacting with the atomic nuclei in the sample. The likelihood of backscattering depends on the density of the atom hit. Heavier atoms reflect more electrons. This allows a computer to produce an image that shows contrast based on density and composition inside the sample. Regions rich in heavier elements appear brighter, while lighter regions appear darker. In biological tissue, this contrast often corresponds to variations in protein concentration, mineralization, or just structural density. Transmitted electrons provide the third viewpoint and are the most essential to three-electron microscopy. These are electrons that pass through the sample rather than bouncing back or being ejected. The amount of electron transmission depends on thickness and density. Dense or thick regions transmit fewer electrons, while thinner or less dense regions allow more to pass through. Capturing transmitted electrons gives scientists a direct view of internal structures without physically slicing the tissue into ultra-thin layers. This is especially valuable for samples that would normally require complex or destructive preparation. Three-electron microscopy brings these three different types of signals together using synchronized detectors and precise alignment algorithms. The microscope records all three electron interactions in real time, then combines them into a single composite image. The beauty of the technique lies in how the data integrates. Surface textures give spatial orientation. Density patterns provide compositional context, and transmitted electrons reveal internal architecture. No single channel dominates. Instead, they reinforce one another to create a balanced, multi-layered representation of the tissue. This data integration solves several challenges that early electron imaging techniques struggled with. Traditional transmission electron microscopy requires a slicing of a specimen into extremely thin sections, often under 100 nanometers thick, way smaller than human hair. That process is delicate, technically demanding, and risks altering or losing important structures. Scanning electron microscopy, by contrast, excels at surface imaging, but cannot directly reveal the internal organization of thick samples. Even systems that combine two electron signals tend to leave ambiguities, because two viewpoints cannot fully clarify spatial relationships within complex tissue. The addition of transmitted electrons allows the microscope to represent both surface and internal features without heavy sectioning, without forcing scientists to infer missing details. The next step into understanding Three-EM is the physics behind how electrons behave in matter. Electrons have wave-particle duality, meaning they can act in one of two ways, either like physical particles that collide with atoms, or like waves that pass through structures, depending on the material's thickness and composition. When the incoming electron beam encounters an atom, the chance of a collision depends on how densely packed the atoms are and how strongly their electrons are bound to the nucleus. Outer electrons are loosely held, so they're easily displaced and become the secondary electrons that show details. Beam electrons that interact with the nuclei are more likely to scatter backward, again creating backscattered electrons. If the material is thin or lightly structured, a portion of the beam passes through, producing transmitted electrons. These different interactions occur naturally in all electron microscopes. The Three-EM technology leverages them deliberately and simultaneously to extract a wider range of information from the same sample. Instrument design plays a critical role as well. The microscope must precisely control the electron beam's focus, energy, and angle of incidence. This alignment causes distortions when the three signals are merged. Specialized detectors isolate each electron type, so their signals do not interfere. Advanced calibration routines compensate for beam drift, charging effects, and slight variations in sample thickness. Modern Three-EM systems also incorporate cryogenic stages that keep biological tissue in a near-native state by freezing it rapidly, preventing dehydration, collapse, or any change over time. Combining this cryopreservation with three-electron detection allows researchers to capture ultra-small details with minimal distortion and with extreme consistency. One of the defining strengths of this technique is the quantitative analysis it enables. Because each signal originates from a distinct physical interaction, scientists can extend precise measurements about the sample, Secondary electrons can be used to estimate surface roughness or topology of the membrane. Backscattered electrons can be analyzed to map variations in composition or density. Transmitted electrons can be processed to calculate thickness gradients or internal layering. When combined computationally, these measurements produce not only images, but three-dimensional representations of tissue, offering insight into how spatial relationships that were previously difficult to interpret can work. Researchers can analyze how cells are oriented, how tightly structures are packed, or how material properties change across a tissue. Beyond that, behind the clean images and elegant diagrams sits a machine that requires extreme precision in every component. Three-electron microscopy is only possible because of engineering solutions that control vibration, temperature, air pressure, and electrical noise to an almost unbelievable degree. Even the smallest vibration from footsteps in a nearby hallway, or a tiny shift in temperature, can change the alignment of electron beams and distort the final sample. All of this also must happen in a vacuum chamber. Electrons can't travel through air without being scattered, so the microscope must maintain a vacuum nearly as empty as outer space. Pumps, seals, and sensors constantly work together to keep the environment stable. The electron source itself also needs to be consistent. Even minor fluctuations in the emission pattern can interfere with the combined signals and reduce image quality. Detector design is also demanding. These detectors must respond quickly and accurately to different types of electron interactions while minimizing noise. Engineers use cooling systems to reduce unwanted background signals and advanced materials that allow for fast readout speeds. All of this mechanical stability, environmental control, and detector precision has to operate perfectly and simultaneously. Without this engineering foundation, the Three-EM technique simply wouldn't function. This technology has a lot of impact, and that impact spans many disciplines. In neuroscience, mapping neural circuits requires simultaneously visualizing axons, which are part of the neural cells, dendrites, another aspect of those cells, and synaptic junctions, and the environment surrounding them. Earlier techniques often force researchers to choose between surface and internal organization. Three-electron imaging can reveal the outer contours of neurons, while also showing how internal components are arranged, making it easier to trace connections over long distances. This is also valuable for studying developmental wiring, understanding neurodegenerative diseases, and comparing healthy versus abnormal neural circuits. Cancer research benefits as well. Early tumors often consist of subtle irregularities that appear not just in cell shape, but in the way cells pack together or interact with their surroundings. Because the Three-EM captures both external and internal features, it can reveal early structural changes in tissue without relying on heavy staining or slicing. This allows researchers to study tumor microenvironments, monitor how cancer cells invade surrounding tissue, and identify early morphological cues associated with malign transformation. In developmental biology, the techniques makes it possible to observe how cells organize into tissues and organs without breaking apart samples. Researchers can visualize gradients, junctions, and branching structures in their natural context. This is particularly valuable for studying embryos, vascular development, and organoid systems, where maintaining structural integrity is crucial. Three-EM enables scientists to observe developmental stages with a completeness that previously required multiple imaging modalities. Material science and bioengineering also gain new advantages. Synthetic biomaterials, engineered tissues, and medical implants often have complex interfaces where biology and non-biological materials meet. Traditional imaging can show either the surface of the material or its inner layers, but not both at once. With three-electron microscopy, researchers can study how cells adhere to scaffolds, how structures behave under stress, and how engineered tissues integrate with host environments. Ecological research sees benefits too. The technique can reveal the minute structural adaptations of organisms like plants, fungi, and invertebrates. It provides a way to compare species with subtle anatomical differences, or to investigate how microscopic structures contribute to survival in specific environments. Being able to observe internal and external features simultaneously makes comparative studies more accurate and less dependent on invasive sample preparation. Another expanding area is clinical pathology. High-resolution, multi-signal imaging of biopsy samples could lead to more accurate diagnoses. Pathologists often rely on stained tissue slides and individual electron microscopy images to identify abnormalities. Three-EM could reduce the need for multiple tests by providing a comprehensive view of surface structure, density patterns, and internal organization within a single image. Faster and more integrated imaging has the potential to improve diagnostic confidence and decrease turnaround time. Beyond immediate applications, the technique opens possibilities for future technologies. Advances in detector sensitivity, faster computation, and machine learning-driven image processing may eventually allow three-electron systems to operate in near real time. Researchers envision automated pipelines where thousands of tissue sections are imaged, aligned, and analyzed with minimal human intervention. In the long term, these developments could contribute to large-scale mapping of organs, identification of early disease markers, and even non-destructive digital reconstruction of biological specimens. Three-electron microscopy also encourages new ways of thinking about biological imaging. Instead of treating surface, density, and internal information as separate datasets, the technique demonstrates the value of integrating complementary signals. This approach may inspire other imaging methods that combine optical, chemical, and physical information. Hybrid technologies that merge electron imaging with other technologies could offer even more detailed pictures of biological complexity. The combination of improved physics, engineering, and computation also makes the method more accessible. Early electron microscopes required highly specialized sample preparation and very long imaging times. Newer three-electron instruments aim to streamline the workflow. Automated alignment, real-time correction algorithms, and intelligent detectors reduce the need for manual adjustments. As the technology continues to mature, more laboratories will be able to use it for routine research, rather than reserving it for rare, high-difficulty projects. Three-electron microscopy is still evolving, and many believe it represents the beginning of a new era in nanoscale imaging. Future versions may include faster detectors, which would allow scientists to watch chemical reactions or cellular processes as they happen. There's also interest in reducing the physical size of these instruments. While today's systems require large, specialized facilities, miniaturized versions could eventually fit on our standard lab benches, making them accessible to far more schools and research groups. Another direction is hybrid imaging, where electron data is combined with optical or X-ray measurements. Merging these could create ultra-comprehensive maps, revealing information that no single method could provide on its own. The scientific impact is significant. As 3EM becomes more common, it will likely influence fields ranging from medicine to climate scientists to materials engineering. Researchers will design more efficient batteries, understand diseases at the molecular level, and create new materials with properties that were previously impossible. By offering such a detailed view of our microscopic world, 3EM has the potential to reshape how humans approach discovery itself. In conclusion, the three-electron microscopy represents a significant leap forward in visualizing the microscopic world. By merging secondary, backscattered, and transmitted electron signals, it produces images that are comprehensive, contextual, and scientifically meaningful. The technique improves structural accuracy, expands quantitative analysis, and opens new possibilities for research in a variety of fields. It's a tool that allows us to not only see structures, but to understand how they work together, to create the systems that sustain life. As the technology continues to evolve, it promises to deepen our insight into the invisible world and to support discoveries that shape the future of science and of medicine.
Thanks for tuning in to the Breakthrough broadcast. If today's conversation sparked a new idea, share the episode and follow the show wherever you listen. Got a question, idea, or breakthrough of your own? Reach out at breakthroughbroadcastconnect@gmail.com. Until next time, keep questioning, keep building, and don't wait for the future. Help create it.